Hi, everyone. Good morning, good afternoon, wherever you are. It's a great pleasure to be here. Thank you, Sarah and Alexi, for having me and for the very generous introduction. And I'm super excited to do this. And especially as Sarah mentioned, the lineup of this virtual seminar series is just fantastic. Please tell your friends, please email Sarah for the links if you don't have it. And I'm very excited to see everyone here. I can see everyone's name and the little avatars, and it's good to meet friends new and old here. Just like perhaps many of you, we are also interested in understanding the neurobiology and the brain through genetic approaches. So I want to just highlight that, summarized by this schematics. Our goal really is to develop technologies and platforms to allow this kind of analysis that I'm showing you here. And we really want to do is to be able to scalably and systematically to perturb candidate genetic factors and being a particular reaching loci gene expression. And specifically, we're very curious about among all the heteros tissue type and cell types, what are the sites of actions for these genetic loci? And within these cells, we also want to ask at whether without this particular perturbation, what are the cellular states changes? Is it possible that some specific subtypes or specific cell states that's more vulnerable to this genetic alteration than others? And within these cells that are vulnerable, what are the cellular changes that downstream targets responding to these perturbations? And most recently, we're also interested in building technology to grow from intrinsic cellular effects that within the perturbed cell itself, but also grow to the neighboring cells to understand how the perturbation can impact the cell itself as well as the cellular neighbors. So this is a summary of the past work as some of the ongoing work that we're developing in my own lab. But I want to mention that the motivation of developing these technology were really facilitated by sort of or inspired by all the technologies as well as the human genetics knowledge that we have gained collectively in the past decades or so. And it's really the accumulation of almost 20 years of work that now we know our vulnerability to many of these devastating disorders, especially neuropsychiatric disorders, actually have genetic basis. So for example, taking the cases of autism spectrum disorder and neurodevelopmental delay. And we actually know that many of the genetic causes or variants that's strongly associated with these disorders. For example, almost 20 years ago, we know that the disruption of a single gene, MECP2, actually leads to a subtype of autism called the Rett syndrome. And in this case, we know that MECP2 encodes a transcriptional modulator that changes how genetic transcription and how the RNAs are made and these processes in the cell. But if we shift our attention to another set of autism risk genes, and in this case, what I'm showing you is a single gene called SYNGAP. And in this case, very different from MECP2, SYNGAP actually encodes a GAP protein that operates on the synapses that regulate receptor trafficking and neuron transmission, essentially how two neurons communicate with each other. So you can think about this is quite different from the cases that I showed you earlier, MECP2 that regulate transcription. So in the past, I want to say 2 decades, there are many of these single gene studies. And I think these are very important foundational work that help build these important models and allow us as a field to collectively use electrophysiology, molecular biology, or even behavior studies to approach this very complex question of autism and neurodevelopmental delay. But when we started a few years ago, our question is sort of slightly different. That is, among all these different autism risk genes, and I know so far we have a lot of progress in identifying these single genetic variants and loss of function and contributing to the manifestation. We wonder how many different kinds there are, and is it possible that each of the hundreds of risk genes actually present an independent question and independent possible underlying mechanism Or is it true that they actually can converge and diverge into subtype? Are there any convergent pathways or convergent cell types of action that we can identify? And I should mention that many of these risk genes actually express quite broadly in the development. So an obvious question is, are these different risk genes actually affect similar cell type or same cell type? And within these cell type, is it possible that they actually converge onto similar molecular pathways? So these are important information to know in order for us to better subtype or even group them together for consideration for therapeutics designs. And a few years ago, again, we decided to focus on a particular subtype of these variants associated with autism spectrum disorder. And these are the de novo and often loss of function protein truncating variants. And these are the variants that you can see, for example, that is present in the proband that affected child, but not in the unaffected family members. So this kind of variants are often highly penetrant that carry the large effect sizes, which is different than the GWAS variants or these kind of polygenic variants that you might be thinking of. So we think these kind of loss of function variants can be an early entry point to understand the biology of this complex disorder. And this is also because in the past decades, there are really a growing knowledge about these de novo variants and really coming from many scientists, many institutes in the US and above, that now we know there's a growing list of these de novo variants contributing to the manifestation of the disorder. And as you can see that about a decade ago, we had less than 10 genes, but in the most recent analysis, I think it just came online in Nature Genetics last month, there are 71 genes that now we know have genome-wide significance that are contributing to the disorder. And this is, I want to mention that the kind of significance we never had in the field for these rare de novo loss of function. So now we know that there's a list of reproducible loss of function variants that's contributing to the disorder. And the question is, are there any common mechanisms among these 71 risk genes? And most importantly, these kind of de novo variants discovery has been fruitful not only for autism, but also for other neuropsychiatric disorders such as bipolar and schizophrenia. So as the gene list keep growing, we really want to have a generalizable approach to understand not just one disorder, but beyond taking a genetic approach to study the mechanism of these devastating disorder by understanding how the risk gene impact our brain development. So a few years ago, we started thinking about this, and we want to establish this system and hopefully a more generalizable system to be able to introduce perturbation in the scalable and programmable way. And we want to do this perturbation in vivo in the developing brain throughout development, because the very nature of these developmental disorder that perturbing in adults will be very limiting in our way of understanding its developmental origin. So we can use CRISPR-Cas9 gene editing system to introduce the guide RNA into the uterus and typically into the developing lateral ventricle to basically perturb the genetic composition of these developing neuroprogenitors or immature neurons. And in the subsequent days or even weeks, these perturbations are happening in vivo before the postnatal harvest and enrichment of these perturbed cells. And we want to use a cell type specific way and high resolution readout to analyze for each of these perturbations what are the associated phenotypes. And the goal of this approach is to be able to test against a large panel of genes rather than doing this one gene at a time. And also we want to gain insights of these individual changes of the cellular actions with a high resolution and ideally single cell resolution to be able to ask how are these different risk genes impact different cell type differently. And I think brain is just famous for its cellular heterogeneity compared to many other tissues, which really presents another layer of complexity in this design. So in order to do this, to study gene function in the development in a scalable way, a few years ago, we really stood on the shoulder of giants who developed in vitro Perturb-seq that was published in 2016. And we built our experiments on the Perturb-seq system and pretty much borrowed the molecular design in this version of the Perturb-seq. So what we can do is to design the lentiviral vectors. In this case, we design 2 guide RNA to targeting the same gene, followed by a fluorescent protein and the barcode. We can inject this lentiviral vector in development in the embryos, in the lateral ventricle of the embryo that expressing the Cas9 protein from the transgenic animal. And we chose lentiviral vector in this case because in development, cells are rapidly dividing. An episomal vector such as AAV will not allow the reliable labeling of the lineage. So you can imagine we perform these injection quite early at embryonic day 12. And in the subsequent days and 2 weeks, that is our chosen time analysis, the lentiviral can target and integrate into the neural progenitor as shown here in the cartoon. And all the cellular progeny in the upcoming days will actually also carry the expression of the fluorophore as well as the loss of function. So at postnatal day 7, what we have is this highly mosaic brain that has many clones of cells, including glial cells and neuronal cells, that's being labeled and perturbed by the vector that we injected initially. So at postnatal day 7, we can disrupt the tissue and dissociate, and using FACS to purify these blue perturbed cells and perform single cell RNA sequencing to capture the isolated single cells transcriptomic profile. And here, importantly, single cell readout not only offer a high resolution approach to capture the phenotype, but it also allows us to multiplex this approach. So instead of injecting one vector at a time for one perturbation at a time, we can actually inject a lentiviral vector pool that each of the different perturbation identities decoded by the barcode that's detectable by being on the poly-A transcript. And we can perform these pool perturbation and only identify each individual gene's perturbation identity at the analysis stage. And ultimately, by performing these kind of scalable perturbation experiments, our question is what are the molecular pathways and cell types that's most vulnerable across this panel of different autism risk genes compared to the internal controls? So there are a few sort of features that we designed into this particular experiment. That is that we diluted our lentiviral vector to make sure most of the cells harvested only have one and only one perturbations. And that's just because the nature of these variants that they don't really co-occur in the patient populations. And it's important to think about that this approach is obviously more scalable because it's more efficient, that allow us to perturb not just one gene at a time, but rather do this in the pool. But I want to argue that precisely because of this, this approach is more well controlled because we can always include a negative control in every single batch of the experiments. And that allow us to build a regression to against all the possible confounds here, such as different sequencing batch, such as developmental timing, subtle differences across different batches that allow us to have a more robust readout of these individual perturbations. And in total, we analyzed 35 risk genes against the control vectors and collected more than 40,000 cells across 17 independent experimental batches. So this experiment was repeated 17 times, that with all the perturbation and guide RNA and the control in each of the batch.
Xin, can I ask a quick or two quick technical questions?
Yeah, no problem.
Do you have an idea of what the transfection efficiency is? So how many cells in a mouse brain or in a given area, what percentage of cells would be targeted? And the second one is, what is the negative control? Is it just a guide RNA that targets something else and doesn't, or what does it do?
Yeah, both are important questions. Thank you for asking that. I will show you a picture for the first question about the in vivo transduction rate. And I think you're right that for any in vivo experiment, the most challenging part is to harvest enough cells from the intact tissue to be able to perform these kind of analysis. And it's obviously something that is titratable. In this particular case, we're kind of choosing a medium range of the transduction rate, because we don't want to have a very high transduction rate so that the phenotype of observing might be coming from the non-cell autonomous phenotype, right? Like it's because you are either neighboring or pre or post-synaptically connected with a cell that receives perturbation, and therefore you present certain transcriptional changes. So we want them to be actually rather sparse and spotty. But this is something that we can obviously change by increasing the concentration of the viral vector that's using, and especially given AAV and lentiviral both can be compatible with this, I think will be interesting for maybe different kind of questions or setup. And for the second question, that is sorry, remind me again.
Was the negative control just what?
Oh, negative control, importantly, yes. So in this particular experiment, we included negative control that is actually a guide RNA that targeting the GFP ROSA 26 loci. So we don't want to identify a convergent pathway being double-stranded break in the genome. In this case, we designed a guide RNA that cuts somewhere in the genome, actually in the transgenic mice, ROSA 26 loci, but hopefully don't really convert any sort of phenotype. But in the future analysis, we're also interested in designing a control in a more biological way to decode sort of what is the autism convergence. So working with human geneticist Stephen Sanders, we've identified basically a list of genes that do express in the development, but has not been shown or in any kind of analysis that confer risk in autism spectrum disorder. So you can think about them as the housekeeping gene equivalent, and apparently these kind of genes are actually rather hard to find. We definitely are interested in doing control, not just for the CRISPR editing purpose, but also helping us to decode, helping us to interpret the data slightly better.
Great, thanks. And we've had a question in the chat as well from Ruben Ya here at Imperial. And he asks, does injection itself cause inflammation to affect the development of the brain?
Yeah, that's a great question. I think it certainly does, just like all the manipulation will change in some way, subtle or not, the cellular properties. So I think comparing the data with the internal negative control is very, very important. And most recently, we're also trying to adapt different kind of viral vector, like for example, AAV are arguably slightly more benign in terms of the inflammatory responses, and then we're interested in comparing what are the differences. But I think these genetic perturbations that we're introducing, first of all, they are only occurring in neuroprogenitor stage, and these lentiviral in vivo, their half-life is rather short. So you can think about this as a transient infection and inflammation. If anything, is a transient response, and all the cellular progeny that's derived from the neuroprogenitors are not being sort of subjected to the persistent inflammation. But that's a really important question to keep in mind.
Great, thank you.
Awesome. Okay, so we were just talking about the general sort of flow of the experiments, and I just want to highlight that there are a few features that actually also built in with this in vivo pipeline, which is that we are definitely analyzing a mixture of homozygous and heterozygous loss of function, because the in vivo CRISPR is hard to control for the specific zygosity. And also, as we're just sort of talking about, it is important to think about that we are actually analyzing cells that is in this conditional knockout kind of sense. They are sitting in the sea of wild type developing normal unperturbed cells. And therefore, what we are focusing is these primarily cell intrinsic or cell autonomous transcriptional effects. And most importantly, we also have to think about we are perturbing the developing brain only from embryonic 12 onwards, and we are not really mimicking these sort of the more traditional germline knockouts, which typically deprive the gene rather early. And in this particular analysis, we'll also only analyze the effect at postnatal day 7, which is quite an early time for brain development. In the future, we're interested in expanding and extending these analyses in later developmental stages to capture some other developmental processes such as synaptogenesis. And we also imagine that this approach can be used in additional organism and tissue types as well. That's not limited to the brain or to mouse specifically. Okay, so before going to single cell data, I'm going to show you some images just to see what this type of experiments look like. So first we noticed that after about 12 hours of injecting the lentiviral vector, we can already observe the expression of the fluorophore in this case. So we're happy to see that, just because during development, if you miss 24 hours, you miss the entire layer of the cortical projection neurons. So we want these Perturb-seq vector to be fast acting, to not miss any cell type that is being dynamically generated in development. And postnatally, I think this is postnatal day 7, we can harvest the brain and look at all these spotty red cells, that is the Perturb-seq vector. And we found that by doing these lateral ventricle sort of early injection, we actually are targeting a really big classes of different neuroprogenitor that give rise to neuronal and glial cells in different brain regions, including the cortex, hippocampus, the thalamus, and so on. So although this study is primarily focused on cortex, but in the future, we can expand this approach to understand these restricting variants and their action in different brain regions, which is very exciting. Okay, so by focusing on the postnatal day 7 cortical tissues, we can isolate these individual cells and perform single cell RNA sequencing. And we can uncover 5 major cell types that we expected at this time of the development from the cortex. So on the right is just the canonical marker expression, and you can see that we can reliably identify these cell type and annotate these cell type to confirm that the identity from our analysis. But I want to highlight that we can also take each of these cardinal cell type and further classify them into subtypes. And for example, in the case of excitatory projection neuron, we can further classify them into subtypes, and these subtypes correspond to their layer positions in the cortex. And you can imagine that our readout of the assay is a single cell resolution, which is that even if one out of these 5 cardinal cell type on the left that has a perturbation effect, or even just a subtype of these numerous subtypes you're seeing on the right that has a perturbation effect, it will still be read out and analyzed and wouldn't be masked as it probably would in the bulk sequencing or any kind of bulk analysis. So our goal is really to identify what are the potential subtype or the cell type of action that each of the perturbation can impact at high resolution. So in terms of the analysis, I often ask this question about how many cells are enough for a given analysis. And I think this obviously depends on many things, but most importantly, depend on analysis of chosen. Very traditionally, I think many of us are familiar with the gene level analysis, such as DE-seq, that allow us to sort of examine 20,000 genes as independent variables. But we also know that the biology in general do not operate on individual genes. They impact gene networks. And we hypothesize that if these perturbations indeed give rise to a certain effect, we expect not just 1 or 2 genes to change its expression, but rather a network of them. So here, I will show you a downsampling analysis from an earlier Perturb-seq work, and to emphasize that why we prefer these gene module or gene program analysis. So this is actually a data set coming from more than 60,000 of dendritic cells upon the perturbation of LPS administration. And in this heat plot, we can start doing subsampling analysis by the x-axis, which is reducing the sequencing depth, or by the y-axis, which is reducing the number of cells included in analysis. And you can see that when we start moving from the top right corner down, the correlation drop quite quickly, meaning that analysis is quite sensitive to the number of cells that included. But for the simple analysis, if we are using the gene module as a readout of the phenotype, you can see that we can subsample to almost 20% of the total cell types, but the phenotype remain quite robust and consistent. So in general, for this kind of gene module and gene program analysis, we can sample as few as 50 cells per perturbation per cardinal cell types, and perhaps sequence a few thousand transcripts. In general, the gene modules can give more robust phenotype, both for our own data sets as well as the published data sets in 2016. But as a biologist, I also want to argue that gene module and gene program approaches are more interpretable as well. So instead of getting 1 or 2 gene that I often have to Google a lot or PubMed a lot to identify what are the possible sort of downstream target that's impacted, and to design better follow-up and validation experiments. And in this case, with gene module, what I'm getting is a list of genes that's already sort of emerged as a pattern or an inherent sort of network that just helped me to come up with potential hypothesis for the validation. So this is what we ended up doing with our data sets, which is that we start with each of the one of the 5 cardinal cell types, and we can learn de novo from our own data sets to look for what are the co-regulated gene network throughout all the cells, regardless of their perturbation identity. And in parallel, we can actually also separate these cells based on their perturbation identity. And by integrating these gene module identified as well as their perturbation identity, we can perform the linear regression and generate the perturbation score for each of the perturbation and ask what are the gene module expression changes with or without the perturbation compared to the control. And across all these different cell types, we extract a total of 14 gene modules across all the perturbation groups. So here, I just want to give you a sense of the kind of module that we can extract it from data sets. I'll take the projection neurons data sets as an example. Sometimes the kind of module we're getting seems to be specific to a subtype. In this case, what I'm showing you is this projection neuron module number 1. And you can see that the expression of this module are highly enriched in the layer 4 population of the projection neurons. And the aficionado in the audience can also notice that some of these genes that are highly ranked in the module are expressed in, are basically the key denominators and determination factor of this layer of the projection neurons. So we call these kind of modules subtype specific. But sometimes what we're extracting are the kinds of module that doesn't seem to be specific to a particular subtype. As you can see, the module number 2 from the projection neuron that seems to be expressed across all the cell types on the feature plots. And in this case, by looking into the genes within this module, we identified that this is actually a module that decodes neurodevelopment, which is a shared feature across all the different projection neurons, regardless of their laminar positions or subtype. So this is the second kind of module that we identify, which is more specific to a biological process or feature, rather than just a subtype. We use the WGCNA, which is a more canonical pathway or a co-regulatory gene network identification tool. We also use the structural topic modeling to orthogonally identify the modules, and both reveal very similar sets of modules, which we are convinced that is capturing certain kind of signals that is quite robust in the data sets. So this is really a bird's eye view summary of the data that each of the perturbation is shown here by column, and each row are basically the 14 gene module that I told you earlier, coming from 5 cardinal cell type labeled here in different colors. So you can see that some of these effects, even by this bird's eye view, are quite cell type specific, such as the perturbation of this gene ADNP that is very specifically impacting just the projection neurons. But some of these perturbations actually hit multiple modules and multiple cell types, including glial cells. And this again highlights the power of the single cell analysis to be able to capture these phenotype and identify identify and match the phenotype to the cell type that they belong. And I also want to mention that previously, since I was trained as a neuroscientist, as Alexi mentioned, I was very sort of excited in doing this analysis to look for neuronal specific phenotype. But to my surprise, there are not only neuron but glial impact onto several of these perturbations, at least at this time of development that we examine. So it's interesting to think about both neurons and glia can be impacted, which is helpful to think about how to better model and analyze these risk genes, especially in vitro when the cellular heterogeneity can be slightly more limited. So, I didn't get to sort of talk about this in detail, but many of these gene modules and signatures are found largely conserved in the human developing brain, which we perform additional analysis to confirm. So these modules that we extracted are the gene expression pattern that also capture in the postmortem human brains as well. And overall, we hope that this method is a scalable and versatile method that can help to identify additional genetic variants in the future and can be applied across different tissue types and particular genetic variants of your interest as well. But as any genetic screen, you might ask me, well, have we really learned from these hits and what does hits really mean? So I will go through some of these key phenotype with a significance before diving into a detailed sets of validation experiments at the end. So what have we really learned? We learned that there are in total 4 cell types that's significantly impacted, that are projection neurons, astrocytes, interneurons, and oligodendrocytes. These are all cell types that also found in the analysis that I'm not showing you here as the work is already published, that we analyzed the human postmortem autism data sets published from Arnold Christian's lab. And these are also the 4 cell types that stand out with the significant differential gene expression. And specifically, just diving into the data, we found out that the layer 4 projection neuron seems to be particularly vulnerable in this time of the development. And it's interesting to think about that this is actually the time that the thalamus, which represents sensory input, being wired into this layer of the cortex. So the changes of gene expression we observe here in layer 4 might be linked with the abnormalities in the sensory processes that many, including David Ginty and Laura Orefici, has reported that the hypersensitivity and potential mechanism linked with the autism patients. And in addition, we identified that in a subtype of interneuron that expressing the NDNF peptide, the perturbation of ENK2 seems to upregulate the expression of a synaptic module and encode the synaptic genes such as potassium channels and GABA receptors. So we suspect that this is related to anchoring's role in regulating neuronal connectivity. Previously, we know that ENK2 has no role in regulating connectivity in excitatory neurons, but this is where we identify its new role in interneuron, although it's in the similar molecular pathways. In addition to the neurons, we also identify there are 2 homeostatic signatures from the protoplasmic astrocytes that seems to be perturbed by several of these perturbations. But in the next few minutes, I will tell you one example of a case that we validated the results in the germline mutant animal. And this is about, again, another glial phenotype called oligodendrocytes. So many of you already know that oligodendrocytes is the glial cell type that deposit myelin onto neurons to not only assure the electrical conductivity, but also help facilitate the refinement of neural circuit during development. So in our data set at postnatal day 7, many of the oligodendrocytes are either rather quite young and immature, and you can see that a majority of the population are actually the progenitor and cycling oligodendrocyte progenitors, and only a small tail that is the mature newly formed oligodendrocytes. So in our analysis, we identified that this particular module, ODC1, that is highly expressed in the progenitor. So you can think about this module as an immature module or progenitorness module. And the perturbation of 2 genes, one being CHDA, significantly decrease this ODC1 module. And this is interesting because CHDA is a chromatin modifier that can recruit histone protein to regulate Wnt signaling in development. And when we just analyzed our data, we found out that there was a paper performing ChIP-seq to identify the direct binding partner of CHDA in the oligodendrocytes and identify that they're actually interacting with motif that's essential for the survival and differentiation of oligodendrocyte, which gave us more confidence of our data. So we move on and actually use the CHDA heterozygous loss of function animal and to quantify the gene expression in situ. We used the two canonical markers. Here I'm only showing you one, PDGFR. That is an immature marker that we identified from our gene module analysis. And using the RNA-seq, we can quantify these gene expression in situ and also allow us to have a little glimpse of the spatial heterogeneity from the oligodendrocyte that is either in the upper layer or deep layer of the cortex. And what we found out is that in both upper and deeper layers, we can identify that a CHDA heterozygous animal present about a 30% reduction of the oligodendrocyte marker, PDGF1. And this is also the similar effect sizes that we observe in the Perturb-seq data sets. So it's interesting to think about that, starting with a risk gene perturbation, we identified that the changes of gene expression in the glial cell type and further characterize this in the germline mutant animal. So just to summarize what I've told you here, I hope I've convinced you that this is an experimental approach that can be generalized to understand genetic risk variance across development. And this is a scalable approach. As you can imagine that we can add in additional perturbation and still perform full perturbation to be able to analyze many risk genes within a single experiment. We have really learned a lot from the single cell analysis. And I'm really grateful to have these high resolution readout to really identify the cell types of action for these individual perturbation that would have been masked by bulk sequencing. As you can see that many of these cell types specific response that we identify are quite specific. And this is really a very rich data sets that we are still learning even after it's published, and many of them can be follow up. But just for one particular phenotype that I briefly described to you, we performed a functional follow-ups that we identified the changes of a glial cell type oligodendrocyte differentiation trajectory might be able to influence its networks. So starting from a pool screen in vivo, we identify a cell type specific effect, and that both neurons and glia are actually directly impacted by this pool of perturbation. And interestingly, we even identify a rare subtype oligodendrocyte that may have been overlooked using the bulk approaches and perform the validation in the germline mutant animals. And I also want to emphasize that here, I think these risk genes are obviously important for its clinical translational power as well as potential to really help understanding the disease pathology. But I also want to argue that these risk genes are actually key entry point to understand fundamental biology and very interesting pieces of biology that we didn't note. Because genes and molecules directly don't give rise to thoughts or behaviors or disorders, because it's really the nervous system that does. So approaches, including functional genomics methods such as Perturb-seq, really allow us to take this list of genes and proteins and going in and taking a systematic approach to understand what subsets of the nervous system and what particular subtype of cells or molecules that they impact on to allow these kind of function as well as dysfunction. So this is probably the most important slides. Most of the work that I described to you were done when I was a junior fellow in Feng and Aviv and Paula's labs. And I was really helped by these two young scientists, Amy and Michelle. Now, both of them are already in grad school. And here is my lab that I'm very proud of. We started last July at Scripps, and we are definitely actively recruiting. If there's any interest in any of the works that I described you today, please reach out. We would really love to have new members to join at all levels, from graduate student, postdoc to technicians. So please reach out, and thank you so much for coming.